I want to welcome all of you who have joined us here physically and those who are across the world. Thank you for connecting in for this special series of clinical trials, series for medtech innovators and researchers. And today we'll be talking about the basics on clinical trial implementation and coordination. And I want to thank you for making it. I am Dr. Twine Daniel, and I'll be fully going through the session of introduction and also to introduce other co-trainers and the team which is online. We really want to appreciate you for joining in. I would encourage all those who have joined in to put their name and their country so that we can know where you come from. That would be very helpful. To read your name and your country. Yeah, so I think just to sort out if you think we have some feedback here, and whether the team online is able to see, you can send me a chat just to be sure you can hear well and there is no problem on your side. Yes, so you can send me a chat just to be sure, just for another double checking. All right. Thank you, Isma. I'm loud and clear. Thank you so much. So you can also go in the chat and give us a bit your introduction, your name and where you're coming from. We really appreciate that. Yeah. So our program today is really starting with with registration. I think some have registered, and those who are online, we already received your registration. We will at one point ask you to open your cameras when we reach at the point of taking a screenshot, and we'll consider that for registration. So we'll encourage you to keep your microphones muted throughout the program, and also to write in the chat, as I said, to tell us something about you. And I'll be introducing shortly my co-trainer, and we'll be able to start right from that point and have a wonderful afternoon. Yeah, so just to start with, we're going to have an introduction of facilitators and also the recognition of the participants. Some are here physically, we are happy to see them, and others are online. And we will also have a short overview of Soil Research Foundation and the CIPHER project under which this training has been prepared. And we'll now dive into the basics of clinical trial implementation and coordination. And we'll also be able to discuss something about the medical device federation. We may not go into that, but I'll be telling you just at the end. And we'll have a session of question and answers just to be able to get your questions. So remember, as we requested you, that if you have a question, you can already send it on the email that was shared with you. And others, we will receive the questions live here at the end of the session. Otherwise, we appreciate your response and participation in this important training. So let me have Susan. Yeah. So as Susan comes, I'm Dr. Twine Daniel, and I am my background, I'm a medical doctor, and then I've specialized in epidemiology, in science and technology, and also in public health methodologies. I am a researcher, I could say, for the last 17 years. Is what I do every day. And I'm in this industry of knowledge generation. I've published over 75 publications and book chapters. I'm a clinical trialist, and I've implemented over 5 trials, and I have a sixth medical device trial ongoing. And I'm also in academia, and I'm involved in teaching for the last 14 years. I support research at postgraduate level, at PhD level, and I'm involved in supervision. I'm also a research trainer, something I'm happy to do because I love sharing the skills that I have received from many places. And we also offer consultants. I'm a consultant, a research consultant. We've supported over 385 research projects. And I'm also involved in biomedical engineering education. Since 2011, we've been promoting biomedical engineering in Africa and starting programs across different universities. And I'm a co-founder, and I'm the chair of the African Biomedical Engineering Consortium, and also part of the African Biomedical Engineering Mobility, which has been offering, we've given 43 scholarships to biomedical engineers to do masters and PhDs and credit-seeking mobilities. So that's a bit of what I'm involved in. I have this intersection between the clinical and also biomedical engineering education, and we've been promoting innovations in Africa as a way of improving the health system and also the economy of the African continent. So briefly, that's what I am. And today is a special day because we are talking about basics in clinical trials, and I invited my friend whom we've worked with for over a decade. Yeah. Susan, good to see you.
Thank you.
Yeah,
I'm
happy
I
need you to
here.
introduce yourself.
Okay. I'm called Susan Barbara Logose, and I'm happy to be here. And like Dr. Daniela said, we have worked together on a number of clinical trials, and this has given us opportunity to gain more experience, and which we would want to share with you today. Thank you.
You need to mention that you are
I'm a clinical trial monitor, which I'm practicing for the past 20 years. Thank you.
All right, thank you. So she's a clinical trial monitor, and she'll be talking to us a bit of why that is important in clinical trials. Yes, so just for the media. No, yeah. So I hope it's not a feedback that goes back to everybody. Yeah. So as they remove that, because I don't want it to interrupt us. Okay. Thank you so much. Yeah, it's still there, so you will try to eliminate it. Yes, so the training goal for maybe I need to maybe mute mine. I don't know whether. Yeah. Should I first mute my volume? Okay. Good. I hope it will be better now. Okay, so I'm trying to share. All right, so I hope it's it's there. Good. So I think we are good to go. So the training goal for this particular training is to address the gap in clinical trial expertise within the medical technology sector by equipping medtech innovators and researchers with the essential skills and knowledge required for the design, implementation, and coordination of clinical trials. So, as we may understand, that we have really worked to stimulate innovation in Africa. But if we had transitioned some kind of devices, we could say class 2 into markets, some of these require clinical trial validation, and they have to go through this very important process. And the innovators keep thinking, and they keep creating wonderful solutions. But at times, if this gap is not solved, then it delays the process from ideation or prototype development to marketing. Yeah. So this is what we want to address. Yeah, I don't know the technical team, if you could help me with the sound. It's feedback. Yes, I have some feedback here. Yeah. Okay. So that's the goal. We want to address that gap of knowledge and skill, and which will be very helpful, especially for the African innovators and all the innovators across the world who are involved in medical device innovation and designing. So to start with, we look at the landscape. So are you help me? I think I still hear the feedback. You will forgive me. All right. I just want to be sure that it's not disturbing people. Yeah. Okay. All right. So I hope it's better now. Okay. All right, thank you so much. I think at least I don't hear myself anymore, so which is good. Yeah. All right, so when you look at the landscape in Africa, Africa has a very unique research and development landscape, and with very clear challenges that we all can relate with. And the first one is, and this is really based on research that people have done, there's limited sensitization and inspiration of young generation of research in research and development careers. So first of all, we are lacking in terms of inspiration and number of people who are involved in research and development. And of course, there are many initiatives which have begun to try to solve this issue and to stimulate this interest. We also have weak research and education systems. So much of what we find at the university is basic training. But there is a lot of applied training which is needed in terms of developing, medical devices outside the kind of knowledge that we are able to get at universities. So there's need to learn more and learn more because technology changes every day, and there's a lot of new things, new knowledge that's being generated every single day. Also, another challenge we see is the low research mentorship and motivation. So few mentors, people who can help the young researchers to move from one stage to another. And at times, it's also the motivation to be involved in innovation, in research, and all these areas. Again, this is a common thing, and it cuts across. There's also low technical expertise among innovators and researchers. And this includes expertise in doing clinical trials, but also keeping up with the advancing technologies and skills that are needed to develop certain, sophisticated medical devices. So other challenges have to do with funding, and funding, when we talk about funding, we talk about by governments and also private sector engagement to invest in research and development. And that puts many of low resource settings in a place of dependence on external funding. And when you're relying on external funding, there could be an element of you not being able to address the real problem that you see in Africa, and you will try to align yourself to what has funding. So it takes away a bit of some independence, and which of course can slow down research and innovation. Another important point is the lack of infrastructure and facilities. We see a number of innovators begin, getting sensitized to innovation and research when they're in universities. But as soon as they leave, they don't have that access to support spaces where they can get access to laboratory and equipment that they could need to build up their new ideas, but also research support that can help them to take their devices through a more robust process of research and under mentorship and under support. So the lack of such infrastructure is a big problem. But not only that, there's also GMP certified facilities, places which have certification that can enable manufacturing of these devices at a large scale. So in many countries, they're not there. And if they are there, they have more of, they're more owned by Western countries and investors. Some end up having to shift their production source from locally, from the low-income countries to, again, back to the high-income countries. And of course, it reverses the advantage that would have wanted to have of developing and manufacturing devices locally and benefit from exports and foreign income. So those are pertinent challenges that face Africa, but also Asia and many other low-income countries. Also, the weak and unclear regulatory framework that can support such research and medical device trials. So these are key challenges that Africa faces. And I want to show you a bit of how it looks like, how we see it in Africa. We still have poor African health research and development status. We know that 15% of the world population live in Africa, but 25% of the global disease is also in Africa. And that means if we have the biggest burden of disease, we should be working harder to solve it and to think about different innovations and also research that can generate new knowledge that can help our local people. But looking at the world's research output, Africa contributes 2% of the world's research output. The knowledge that is used in the world, whether in healthcare, whether in industry, Africa is contributing 2%. So little knowledge is generated from here, meaning we also import knowledge. We also import knowledge, and our practices are not based on knowledge we generate locally, but it's based on knowledge generated in different settings. And of course, there could be some bit of disjunction in that. And look at human resource. Health researchers, people who are researching in the lines of health, 15% of the global number come from Africa, very few. And 20 per million, so we normally measure these based on per million population. So 20 health researchers per million population. That is the number for Africa. Compare it with Europe, which is 244 per million people in Europe. A very big difference. And that means that the human resource is lacking, the outputs of research and innovation is lacking. Come down to our country, Uganda. For those who are in Uganda here, we have 6 health researchers per million. Now, very low. We talk about full-time. So we are not generating enough knowledge to guide our decision-making here in Africa, but also in low-income settings. Another statistics, just for looking at Uganda, is just the same, and it cuts across many countries in Uganda. In Africa. So, for example, researchers, general researchers, not just health, per million inhabitants, we have 26.5. Yeah, I don't know are we okay? Yes. Yes, so the team had gone off here. Okay, so we have 26.5 per million inhabitants for Uganda. If you compare it with the European Union, which has 4,369, is just a far cry. We are still very far. We are still very, very far. So looking at these numbers, look at patents and patent applications. When we look at patent applications as per the percentage of the global patent applications, Africa has 0.5. We have not even reached 1%. We have not even reached 1% of the global patents. We've not reached there. So looking at such a scenario, yeah, just allow me to share this. Yeah. Okay. All right. So all right. Yeah. Okay. I just wanted to connect in. Sample had gone off. So when we look at the patents, we have a very low patent rate, and less than 1%. Meaning, in terms of innovations, we are still not doing enough. And it is something that we have to make sure that we change. It's something we have to make sure that we change. At a continent level, we have aspirations, like the African Union Agenda 2063. And the first aspiration is a prosperous Africa based on inclusive growth and sustainable development. Now, under this goal, 1.2, we are looking at a well-educated citizens and skills revolution that is underpinned by science, technology, and innovation. So there is already a framework that should support stimulation of innovation and science and technology in the African continent, at least in line with the aspiration of the African Union 2063. So that is where, building capacities of researchers comes in, and that's why we need to build the capacity of innovators in different ways so that we can achieve these goals. The Sustainable Development Goals still supports, and when we look at Sustainable Development Goal 3 and 9 and Target 5, it's promoting enhancing scientific research, upgrading technological capabilities of industrial sectors in all countries, and in particular developing countries. By 2030, we're about to reach there. And there's a clear focus on encouraging innovation and substantially increasing the number of research and development workers per 1 million people. So all these frameworks are pointing us to the need to do something in line with research and innovation and building capacities of researchers and innovators within the low-income settings to be able to beat up that gap that I showed us in terms of scientific production and innovation contribution. So I would want to briefly tell you about Soil Research Foundation, and in line with how we connect to these key goals, and then after that we shall jump into the clinical trials. So as Soil Research Foundation, our focus is to promote research excellence in Africa, and our approach is to create one-stop centers for health research, training, and innovation services. So as we saw that that's a big gap in terms of infrastructures that could support researchers both inside the university and outside, those who are outside institution of higher learning, to be able to pull up their ideas and push them to the end and be able to generate the knowledge that we all need. So back in the days in 2010, this is the ideology that we started off Soil Research Foundation. And I remember I was coming from my first master's in Belgium in 2010. And what was in my mind was the health-related challenges of Africa are unique and can best be identified and sustainably addressed by equipped and motivated Africans themselves. And something very peculiar with this, and which I really believe even today, is that we can actually see our problems easily. We only need to have trained eyes. We only need to have trained eyes. We can see the problems we have, and if we are equipped with the knowledge of how to solve them, we can actually solve them as long as we are motivated. So this ideology is what we based on to establish Soil Research Foundation, and it is an institution or an African initiative by Africans and for Africans. So we are looking at African people, trying to improve their lives, especially the vulnerable, by promoting, better and high quality knowledge generation processes. That's research and innovation. We are a mission-oriented contract research organization, meaning we can take on research, we initiate research, and we can implement research. We also have research consultants and training services under which this kind of training falls that are our self-sustaining mechanisms. Of course, many entities begin in Africa, especially when they're African initiatives, and if they don't think about sustainability, then they fail. But we've been able to manage because we thought early about a self-sustaining model, and that's what we use. We at a pilot phase to assess the operational model of Soil Research Foundation and how it can contribute to the research landscape that I showed you. And we have a focus on promoting research excellence, particularly in areas that are related to health. And someone may say, but I'm not in health, I'm not, and those are humans. But I'll tell you that when we talk about health or human health, humanity lies at the center of all research efforts. Sometimes I meet people who are engineers and they don't see how their work is relating to human health. But humanity is at the center of all research efforts. Whether you are dealing with water, you're dealing with animals, you're dealing with plants, the end of the story is, are humans being affected? Are you solving a human problem? Because all nature is there to create an environment for human beings to live comfortably and safely. So think about a human being. And that's why whatever you develop, whether it's a medical technology, we would want to see how it fits into a human being, how it interacts with a human being, and how it can improve the lives of human beings. So humanity is very important. So we focus on human. And then health is the major cause of human crisis and impoverishment in Africa. Two things that health takes away. If there's bad health, there will be human crisis people are worrying about their lives and their safety and livelihood. And the next thing is that when there is poor health, people will lose money. They will lose money. And another thing, the productive population will be weakened. It will reduce. So health in human is a key point, and that's what we focus on. In summary, our strategic plan, strategic approach as Soil Research Foundation is to establish one-stop centers, as I said, for these health research and training and innovation services. And we do it by creating reliable, and that's our mission, reliable and efficient infrastructure for quality research implementation, research consultants, and skills development. And our model is more like a battery. And we have these aspects within a one-stop center. We have research centers. I heard the slides are not moving online. Okay, I tried to reshare. Perhaps it will be okay. Sorry about that. Okay. Are we able to? Are they moving now? Yes? Okay, thank you so much. Yeah, so our focus is to really create reliable and efficient infrastructure for quality research, implementation, and research consultants and skills development. As we saw, there's need for skills, and there's need to empowering African researchers and innovators. There's need to build that equipped community of elites who can identify problems and solve them. So in our one-stop center, we have research centers, research training centers, research consultants centers, and research grant management and administration. We also have the Global Health Researchers Mobility Desk. It's a very important desk because some people come from Europe, they come from different countries, and they want to do research, but in Africa. So they should have a place where they can be able to be supported to do exactly that. And we offer that, and also we engage into knowledge management and writing of research books and production. And we think with such a format of a one-stop center, we are able to offer high quality and reliable and accessible research services to support researchers and innovators in low-resource settings, including those who are outside the institution of higher learning. And that's a group which is normally lost. You do a very nice project, you're interested when you're in the university, and then you move out, and you have no support system, and you have nowhere to lean to be able to run your ideas to the end. So through this, we're able to envision research excellence in Africa by improving those R&D indicators that we talked about. And so our objectives are as such: we teach, we support, and we do research. And we have been able to establish a research center, a clinical trial center right here, and we're implementing medical device trials, and we have built a capacity, and we have a very ready team and policies that allow us to do so. And we have also a research training center where we are right now, and which can sit 140 to 150 people. It's open to researchers to come and do the trainings of their teams, but also to receive trainings. And we have all these elements we have talked about up, and you can be able to check our website and see more. So one important initiative that we began is the support initiative for African researchers, which is called CIPHER. And here we're looking at building a big community of researchers in Africa, but who are problem solvers. The theme is be a problem solver, be a researcher. Research and innovation is about solving human problems, and that has to be at the heart of anyone who wants to do research or innovation. It should be focused on solving the problem of the vulnerable in these settings. And I think that's what I can say on that. And the big need is we want to get more human resource that is equipped, and so that it can be able to address these issues. So with that, I think I have been able to tell you about what SOA Research Foundation does. And under the context of support initiative for African researchers is where these public lectures are. And we began last year, and we felt we should have another one this time. And so today we'll be talking about the basics on clinical trial implementation and coordination. If you have any question, you'll be able to ask it at the end and as we go on. In case the slides are not moving, please just alert us and we'll be able to solve it. Otherwise, for those who have just joined us, you're welcome. So let's start. Under this session, we're going to talk about the principles of clinical trials. And we're going to start by introducing clinical research, because someone here may be a beginner and you have not really been part of even the basic of research. And I'll mention just a few things and also give you an overview of clinical trials and the types of clinical trials and introduction to international standards that guide the implementation of clinical trials. So, as a way to start, when you look at research, I want you to think this way. We are looking for a solution for human problems. We are looking for a solution for human problems. That's the heart of a researcher. It is to find solution for a human problem. Whether the problem is caused by bad water, whether it's caused by animals, whether it's caused by weather, whatever you're doing, it ends up with human survival. Human survival. No matter which way we showed ourselves up here, some of us will believe we are created by God. Others believe they came through whatever. I don't know what it is. But look, we found ourselves here, and every day we work to survive. We work to survive. When it is too hot, we cry. We think about light clothes. We think about water. We think about, all that. So we are affected. It's humanity that is affected by everything else, by everything else. When the animals die, when the plants die, we are affected. So when we are doing research, we are looking for ways of solving human problems. And that is at the heart of a researcher or an innovator. Okay? And that will tell you why we need to test what we have developed on human beings to be sure that it's working. So, clinical research can be biomedical or behavioral research. And these are studies which involve human participants. Everyone we say clinical, we mean human. You can't say I'm doing a clinical trial on animals. No, that is preclinical. So clinical means human. We are doing something among human participants. And clinical research has two large kingdoms. These kingdoms, what are they? We have the experimental research and the observational research. So these are two kingdoms, experimental and observation. And the difference in these lies in whether the investigator is assigning the exposure. So meaning, if you are going to give me a drug or you're going to put a device on me, you are the one who is introducing that exposure. So if you're introducing that exposure, it's an experimental study. But if you are just observing me to see what happens because I've had a lot of X-ray, I've gone through X-rays, and what might happen to my health over time, you are not the one who exposed me to it. You are just observing. That's an observational study. So interventional study is the investigator exposes the person to an intervention. While observation, the investigator does not expose. They just observe whether someone has been exposed or they have not been exposed and try to compare what are the outcomes. So in simple terms, that framework you see there, it shows you the process, that this design are dependent on what the investigator is doing. Did the investigator assign exposures? Yes, that's experimental. If no, that's observational study. And next among the experimental study was where people randomly allocated to whether to get exposed or not to be exposed, then that becomes a randomized experiment. It's a randomized clinical trial. So is it randomized? Okay, so we are going to see different types of clinical trials, but that is the simplest in terms of basic understanding of what the two kingdoms are, experimental and observation. Now, research has different levels of evidence, and the lowest is opinions of authorities, like someone telling you, for me, according to me, as a professor, when I look at this sputum, this must be TB. This person must be having what? TB. So that's expert opinion. Expert opinion is the lowest level of evidence. Now, I want you to understand that whatever research we are doing, at whatever level of evidence, it is ending in how that knowledge can affect human beings. So at the end of the day, we want to see how to get what we have learned from research and from innovation into services that can help people, or into real solutions that are going to help human beings. So the level of research matters because it gives us confidence. It gives us confidence. Some levels of research are still low, that they can't give us a strong evidence for us to use that kind of results from a study in a public arena. So when you look at the levels of evidence and the strength of evidence, the highest is systematic review and meta-analysis of what? Of randomized controlled trials. In line with studies designs themselves, the randomized controlled trial is the highest evidence. It's the strongest evidence. And many of the interventions we develop, we want to expose them to this highest evidence, which decision makers and healthcare planners and you could say like World Health Organization use, and they grade them that way. So if you have done clinical trials on something in different contexts, especially if clinical trials have been done in different populations, at the end, we do a systematic review and meta-analysis which tries to bring together all the evidence from these trials and tries to calculate a simple statistic, which we call a meta-analysis, a simple statistic, a pooled statistic that assumes a bigger study that includes all participants that happened in those individual trials. So if one trial had 100 people, and another one had 200, and another one had 300, if we are bringing those trials together to do a meta-analysis, we will assume a population of 600. And that gives us a simple summary statistic, a simple outcome of which shows the effect of that intervention on human beings. So because it brings together already high evidence or strong designs, it is the highest level of evidence. So that pyramid, normally we use it to demonstrate, and clearly so, it shows that the highest level of evidence is systematic reviews. And in terms of studies, it will be the randomized controlled trials. So I hope that is clear. So just to go into the overview of clinical trials. Now, as we saw, clinical trials, they follow under the kingdom of intervention studies, meaning you're testing on someone something you have developed. You're testing it on human beings. You're testing with a drug, you're testing a device, you're testing a diagnostic method. So let's first see what are clinical trials. Now the definition varies. There is a regulatory definition, meaning what Food and Drug Administration, FDA, describes as a clinical trial. In Europe, It is not FDA, it is EMA, which is European Medical Agency. What does it describe a clinical trial to be? And also we have the ICH, which is the International Conference of Harmonization, which I'll talk about during the presentation, which came as a guide that was put together by all countries. Basically, it's a global direction. It's a global guidance. So with the regulatory definition, a clinical trial is any investigation in human subjects intended to discover or verify the clinical, it can be a clinical, pharmacological, or other pharmacodynamic effects of an investigational product. So, and or identify any adverse reactions to the investigational products, and or to study absorption, distribution, metabolism, and excretion of investigational products with the object of ascertaining its safety and efficacy. Just to simplify, you have a device, you have a drug, you have something you want to test on human beings, you want to test how it reacts with the human being, or how the body system of the human being reacts with it, or you want to assess the outcomes of using it, whether it is on improving clinical outcomes, like someone has a disease, they're going to be cured, or you're going to diagnose whatever clinical outcome it is, or you want to assess how it reacts with the body itself, how it's metabolized or broken down, and you want to ascertain two things. Is it safe? Is it efficacious? Efficacious, it means that is it achieving its purpose of addressing the clinical problem or the human problem for which it was designed. So these are very key areas. And when we talk about investigational product, it can be in a pharmaceutical form, meaning it can be a medicine. It is a pharmaceutical form of an active ingredient. At times it can be a placebo, meaning it doesn't have any active ingredient. And it can be a device that you've never tested in human beings, but you want to test it now. All that is an investigational product. So when I talk about investigational product, that's what I mean. World Health Organization also defines clinical trials as, especially for purpose of registration, when you want to register that trial. A clinical trial is any research study that prospectively assigns human participants or groups of humans to one or more health-related interventions to evaluate the effects on health outcomes. Now, what you pick there is that you can prospectively, like, assign someone, an individual, to have a certain intervention, and another individual not to have. But also you can assign an intervention to a group. Let's say you want to assess an effect of adding iodine in salt. And see the health issues that you want to change. Now, salt is used by almost everybody. So you can decide that on the salt that goes to Uganda, you add iodine. And the salt which is going to Kenya, you don't put what? That's if you have taken a country as a cluster, as a group. You can try it on a school. One school versus another. So meaning everyone in that school would have to receive that intervention. And everyone in the other control school, they will have no intervention. But they may have something which looks like iodine. Salt. But they will not, that salt will be looking like this one, but they want to be iodine. And then at the end, you are assessing a certain health outcome. So at times, we randomize to individuals and allocate them to one group or another, or we randomize a group of populations and put them in different groups. NIH also has its trial definition. There are quite a number, and it says a research study is which one or more human subjects are prospectively assigned to one or more interventions. This also looks similar to the WHO. So what I need to mention here is that clinical trials may also be referred to as interventional trials. And these interventions are not restricted to drugs. I know the pharma industry has been here for a long time, and it has been growing since the days of discovering penicillin. They have been growing. So a lot of experience has been built from the clinical trials that have to do with drugs. But also the medical device industry is growing now. And it's going to pick a lot from the experience that has come from the pharmaceutical trials. But when we talk about interventions, I want you to remember that it's not only drugs. They can be cells. They can be other biological products. They can be surgical procedures. They can be radiological procedures like X-rays, CT scans, MRIs. It can be devices. It can be behavioral treatment. That some people, you put them on a routine of physical exercise. And others, you don't put them on a routine of physical exercise. And that's an intervention because you are moderating it. And it can be a process of care. That some people, they receive their care through mobile phone, while others, they go to the clinic. Do you get the difference? So there you've just changed the way how care is delivered. But that is an intervention too. And there is also preventive care. Sometimes you're giving people an intervention which is protecting them. Like elements, head gears. It can be masks. All right? It can be family planning methods like condoms and all that. All those are biomedical devices. They are all biomedical materials. So, all those need to go through a process. And some key words that I want you to keep in your mind as we move on is experimental unit. What is the experimental unit? You must know whom you are randomizing. Whom are you dealing with? An individual or a group? An individual or a group? As I said, you can deal with an individuals and you allocate them to different interventions, or you can deal with groups. And what's an intervention? Intervention has to do with manipulation of a subject or a subject's environment. You can do something to change something on them, or changing their environment for the purpose of modifying one or more health-related biomedical or behavioral processes. So it matters which intervention you're dealing with, but what we call an intervention is that you are intervening if you put something on me, right? All right? You are intervening if you change my environment. You are intervening because you're going to change the way I'm going to live. So, that's very important. And another term you're going to hear me talk about is the prospectively assigned. Prospectively assigned. We don't do clinical trials in the backwards. There is follow-up. If we have exposed someone to something, we need to follow them up. So that's what we call prospective. So we have to follow them up for some period of time that is clinically meaningful, that we are able to ascertain the outcome we are looking for. So that is very important. Another terminology we're going to hear is health-related biomedical or behavioral outcome. So there is always a pre-specified goal or conditions that reflect the effect of an intervention you want to test. If you have developed a device that is helping to monitor the blood glucose levels, glucose levels in blood. Now, we know there will be some existing standard of care devices. There will be others. There are other ways how we can test that blood glucose level. But now you're saying, well, I think the other one is so invasive, I can just put something on my hand without pricking anybody, and it will tell me what blood, the levels of blood glucose. It's okay, yes, that is exciting. We will all be able to test ourselves actually with that because we fear the pain. Okay, so, but if you're going to test it in a clinical trial, that means you're going to randomize two groups, maybe individuals, and you give one this and another one, you test them using the standard of care. The end of the day that we want that outcome. What is going to be the outcome of interest? The outcome of interest is to see whether this one is picking the right concentration of glucose in blood as compared to the standard of care. So if these two show that there is really agreement, then we will say, okay, the efficacy is good. It achieves the purpose very well. But then we will think about something else. Does it carry some safety issues? We also need to compare the safety. Because when we use this, okay, someone's going to get pain. But now this one, it burns the skin. It predisposes to cancer of the skin. Now, we have now to wait to weigh the two. Not only based on efficacy, but also based on safety. Okay? So, we are looking at things like certain outcomes like positive or negative changes to physiological or biological parameters. It can be that. It can be positive or negative changes to psychological or neurodevelopment parameters, or changes in the disease processes, or changes in health-related behaviors. So, quality of life. When you're doing trials in cancer patients whom you know they are terminal, so it's palliative care interventions. So you are improving quality of life. So that is what you're measuring. So that's very important, because everyone who designs a device, they tell us there's a problem they want to solve, and it's from that problem that we get the outcome we need to measure if we are testing that device. So randomized controlled trials, just to introduce them. And this actually approximates to what the people who are in the lab call the controlled experiment of basic science, where you go and you have controlled the environment in the test tubes in the lab, and everything is really controlled. So now that is really the key thing. Yeah. So when you talk about randomized controlled trials, they approximate to such an experiments. But you know it's not easy to control human being, like how you control things in the lab. Yeah, because we change, we interact, our body system changes. Today we are happy, tomorrow we are not happy. So it's not the same, but we try to create a similar kind of environment in clinical trials that can bring us closer to a controlled environment. So the key point in randomized clinical trials is to really ensure that assignment of participants to any exposure, to any intervention or non-intervention is purely by chance. That there is no element of bias that this person, I want them to receive the intervention, while this one I don't want them to receive. No, it should be completed by chance. Just like I would do lots and we say pick. So it should be by chance. If there's any element of that someone has influenced someone to be in a certain group or another, then there's a problem. Okay? So, randomized control trials, they reduce the likelihood of bias in determination of the outcomes. And that's what makes them the strongest evidence, because you're randomizing by chance. Everyone has the same chance of being either receiving the intervention or not receiving the intervention. And that eliminates bias. That means whatever effect we will observe, it will be entirely because of the intervention. And in that randomization process, the differences between people are already distributed equally between the groups. So the outcome we shall find at the end will always be related to the intervention. So that makes the randomized controlled trials very important. And when they are properly implemented and there is random allocation, it helps to eliminate selection bias. And we could also include in randomized controlled trials blinding. Sometimes we blind people. Blinding is not to kill their eyes, but it is to make sure they don't know what intervention they have received. The patient may not know. Or we make things to look alike. But one has an active ingredient, like if it's medicine, it has something to cure, while another one is not. It's bulk, but it looks exactly like. Of course, there are intervention trials where we can't achieve that. Like many times when I deal with devices, because someone will say that you put a device on them, and another one you've put a different device. So you can't blind them. They know what it is. So there we do open label, randomized controlled trials. But there are times when we can be able to do a double blind. A double blind further eliminates bias, and it becomes much more stronger than the open label. And another thing is confounding bias is also eliminated by randomized controlled trials, because we are able to balance both the known and the unknown confounders, things which can explain the outcome besides the intervention you have given. There are some elements or factors that can actually explain the outcome when irrespective of your intervention. So for us to eliminate that, we want to make sure that they are balanced on both groups. Both the group we are giving an intervention and the group which is a control or the comparison group. The confounders should be balanced. To achieve that, we do randomization. We randomly allocate participants to either groups. So that is the beauty behind randomized controlled trials. But they also have, in as much as they are strongest evidence or designs, they also have drawbacks. Like their external validity issues. Meaning when we're doing clinical trials, randomized clinical trials, we try to restrict. We put eligibility criteria. You qualify, you don't what? Qualify. We don't want certain people. You are pregnant. You are breastfeeding. You are not psychologically stable. So you may not understand our consent. So, and yet in the real practice, when the drug comes out, it is used by everyone. So what happens? The effect we see in a clinical trial, it reduces when the drug or the device is now put into the market. So, and that's why we have what we call post-marketing trials. Why? To check a bit to say, hey, the things we found in the clinical trials, are they still the same? Are they still the same? Is it still working? Now, in the real market and the population, you may find that the other group, which you never included in clinical trials, is the biggest. Let's say the trial was done in Europe. Now, the drug is being used in Africa. So you may find that totally, the population of Africa was not included. So an intervention which works well in Europe may fail to work well or to produce at least closer and similar results when it comes to Africa, because of the diversity in Africa, because of the differences. They can be genetical, they can be lifestyle, the way Africans live, right? Something may not work. Something may not just work. So, there's an issue of external validity that when we're not able to really have everybody to participate in a randomized clinical trial, so when it goes out, it might give some differences, some results, although We have published and we've said it's working. And unlike observational studies, the randomized control trials, they include only volunteers who pass through a screening process before inclusion. We want to make sure your liver is working. We are going to give you a drug, but and you're a drunkard. You must be drinking, eh? So if you're drinking, we need to first check you. And yet in the normal practice, of course, all those people will have to access that intervention, and we will never know how it works on them. So those are the limits of randomized clinical trials. And they can't be used in some instances. Since intentional exposure to harmful substance, e.g. toxins, bacteria, or what may be unethical. Like you may discover an organism and say, I want to see if it causes disease. Really? Are you going to expose it to someone? Eh? So, sometimes we can't use them in some contexts because of ethical issues. Then randomized control trials can also be prohibitively expensive. So that's another thing. They are expensive because there are many things to put into control. There are many eyes looking at the randomized control trial. Not only the ethics committees, not only the investigators, not only the government. The government has a governments have a role of overseeing the safety of their population. So they make sure that they are looking there. The government agencies are looking there to monitor what is going on in a clinical trial. There are also international bodies, FDA, EMA, all these, they are watching. And if this intervention is to be passed out on market, there has to be, a way of confidence that everyone has with regard to the way the clinical trial was implemented. Sometimes we have trials where the European Union can come after 15 years. The auditors come. FDA can send inspectors to check on a trial which was done 15 years ago, just to cross-check, was it done properly? How come in other settings we are seeing different results? Huh? And that's why, you don't just do a clinical trial and throw away the documents. We keep them. We archive them. Because at any time, someone and different bodies need to scrutinize the process. They not only looking at a publication and say, oh, the result is this one. No. The process matters much more as the results matter. And of course, look, you're developing a device and you're competing with me who has also my device already in the market. And you bring your device, you think I will not investigate you. The battle at the marketplace will be on, They need to go through a proper process. Remember, you're dealing with giants, those who have already monopolized. So they have the money. And their money is to pull you down. So, and where they will catch you is if this device didn't go through a very robust process. And of course, even if it's registered, a device or a drug can be delicensed. Delicensed. Yeah. So we have to remember that, and that's why we think these skills are very important, that the innovators understand them and understand this process. So randomized control trials may miss out long-term effects because times we can't follow up people for a very long time. So, and yet, when something is in the market, it can be used for a long time, and people can have effects after 10 years, 5 years, and yet the trial took only 6 months. Yeah. Classifying clinical trials based on primary goals and number of participants. So when we look at, let's say it's a drug, it's a chemical, it's a what, before I go to devices, we have what we call the preclinical phase. Preclinical phase, you're testing on non-human subjects. It can be rats, it can be monkeys, and you're interested in the preclinical phase as you're testing this that you're dealing with. It can be a topical device, it can be a device, it can be a chemical, a skincare, it can be a drug, but you are testing it in non-human participants. You want to assess the highest dosing. You want to assess toxicity. You want to assess pharmacokinetics, how this drug is broken down or metabolized, and how much of it will reach the final target, places where it's going to cause effect. You want to assess carcinogenicity, risk of cancer. You want to assess efficacy. Does it work? So meaning you simulate a certain situation in a rat, a behavior. You simulate it or induce something in the animal that will allow you to measure that effect. So there are many things which can be done at this point. I think with devices, there's also bench testing and all this stuff. We'll talk about the devices. Then we have phase 0, where we are doing preliminary evaluation of efficacy, effectiveness, and safety in health volunteers. So meaning you're testing something, a drug on healthy volunteers. What are you looking for? It's the first time in humans. So it's a first clinical trial phase. And you want to know whether it has some effects on the outcome you're looking for and whether it is safe. So you want to monitor a bit that. And phase 1, you're still evaluating efficacy and effectiveness and safety in health volunteers, but increasing numbers. So sometimes you can do 0 and 1 or do 1 and cover for both and do a good number and do like 20 to 100 participants. So phase 2, you are dealing with real patients, your target group, in whom you want to cause a difference. And you are assessing, again, efficacy, and you may assess effectiveness. Why I put effectiveness here is because when it comes to behavioral interventions, we talk about effectiveness, and especially dealing with groups, and safety in patients. So safety and efficacy, they keep going together. Is it working? But is it working without causing problems? Phase 3, as we go on, the numbers increase. And of course, this tells you that the cost of trials also keep increasing with phases that you're doing. But by the time you reach phase 3, you are at the point of after phase 3, the results are going to be used to change policy. They're going to guide guidelines. So we try to increase the participation by minimizing the eligibility criteria. We allow more different group of people to participate in a trial, as close to how it would be in a pragmatic setting. And that way we also go for more people. And we follow them up for some longer time to catch some, effects of the intervention that may happen after a longer period. And after we have tested our outcome in phase 3, then we are able to, you go through the licensing process and marketing process. So let's say you succeed, you've succeeded, your drug, your intervention is now out there, it's accepted, and policymakers have taken it up. So they are going to roll it out and implement it in pragmatic settings. And what's going to happen? It's going to be used. So what you're going to do is we still need to do some phase 4 over time to do post-marketing surveillance to see, are there some new adverse events we didn't find in trials that are coming up? Is the effect still good, or the effect has reduced? When we come to devices clinical trials, medical device clinical trials, the differences are there, but they're not very different from the clinical trials. But we still have the preclinical phase. So on the first column, we have the pharmaceutical trials. And then in the second one, the medical device trial stages. So where we have a preclinical in drugs, we still have a preclinical testing for devices, where we do bench testing, you may do technical testing, computer simulations. You may also do animal testing. Use some models. And this helps you to test your prototype, which you have developed, to ensure that the final product has the safest and most effective design before testing in human beings. So this is always needed when you're starting the regulatory process. If you're going to do a clinical trial, that background is needed. That data is needed. So you have to know that you need to document that. And phase 0 also is what we normally in devices take as stage 0. And it's same goal as pharmaceutical trials. This is the only point at which medical device trials will often enroll as many more subjects than pharmaceutical trials, because here with devices you can take more than 20 in that phase of first time you are testing in human beings. You can move at least to 30, you can go above, but when it is with drugs, because they go inside, we restrict to should be 20 should be very few. You can't expose many people to the drug. And of course this you can do in the health volunteers. Then where we have phase 1 and phase 2 in drugs, in devices, medical devices, we have what we call the traditional feasibility stage. Traditional feasibility stage. When you hear that in the medical device language trials, we are meaning phase 1, phase 2 of the pharmaceutical. So here we are assessing again safety and efficacy of a nearly finalized product only in patients with indication of interest, meaning they have a condition that you are interested in. So you're testing only those ones. You're exposing them to that. And the device at this time, it should be really, really at almost final. It's really final stage. Yeah. And phase 3 is what we call the pivotal stage in medical device trials. And it is same as pharma trials. Of course, they still have bigger numbers. And as we can see that the differences could come in that drug trials, they implement blinding, as I said, blinding of the investigator and placebos. But device trials often only, they are controlled with a group that does not receive the intervention or standard of care. So you can't hide a device that you have put a device on me and I don't know, unless if I'm unconscious. So, so it is difficult there. And we also can do a post-marketing surveillance after a device has been put out. Cluster randomized trials, we talked about these, how they differ from the randomized control trials is because now you're giving the intervention to groups rather than individuals. Types of clinical trials. There are types, I'll mention a few things here, just because of time. Types of clinical trials. When talking about types of clinical trials, they are according to the type of intervention you're dealing with. You can do a treatment trial, meaning you're testing experimental treatments. You can do a diagnostic trial, meaning you're testing you're looking for the better test or procedure for diagnosing a particular condition. You can do a quality of life trials, like these are supportive care trials. You're helping someone. You're developing processes. You are giving someone a 3D, or a virtual reality kind of innovation so that you can help them to work their paralyzed hands or legs. You are developing something to help someone to improve their quality of life irrespective of the condition they have. So there are also screening trials when you're dealing with screening tests, and also prevention trials. So you have to know what you're dealing with. Another group is based on the stage of development. So it can be phase 1. You can call them, this is a phase 1 trial, or it can be a therapeutic exploratory trial like phase 2, and so. So as I had showed you that different phases will measure different things, and at time when you're naming your trial, you need to name it also according to the stage of development, whether it's a phase 1, stage 0, or whatever stage. Sometimes we describe trials based on, again, stage of development. Is it on therapeutic confirmatory? Are we in phase 3? Are we confirming efficacy of the device? Or are we in post-marketing phase? So we can label them like so. Sometimes we have to add another description on a trial that has to do with the study design. Is it controlled trial, meaning there is a control group and we're going to compare? Is it random assignment of whether a person is going to be on this arm or this one? Is it random? Sometimes it's not a randomized trial. It can be a quasi-experiment, meaning it's not randomized. The assignment of participants in each intervention, whether intervention or control group, is not random. So that could be called a quasi-experiment. Blinding, if there's blinding, you want to mention whether this is a blinded trial. Sequence, you want to mention whether it's parallel, meaning there are two groups. You give this one an intervention, you give this one a standard of care. Or is it a crossover? You get this, and then there is a washout time, and then we give you another intervention. Okay? Or is it factorial clinical trial, where we are not testing only one intervention, we can test 2 different interventions in the same participants. And hypothesis, also want to know what hypothesis are you dealing with. Is it a superiority clinical trial? Are you looking for something that is better than what is existent, that is superior? Or are you dealing with a non-inferior clinical trial, meaning you don't mind, you want to check whether it's just as good. So if it's as good, it could work as an alternative or an adjunct, something to add on. So all these help us to categorize clinical trials. And whether it's individual or cluster trials, are we randomizing individuals or are we randomizing groups? Groups are clusters. As I told you, schools, churches, countries, villages, or we are dealing with individuals. Multi-center trials. We again also think about describe trials according to whether it is done in one center or in many centers. So if there are many centers, you hear something like multi-center trials. They all have their dynamics of how to manage them. Now here we have international standards. So research is done, it has to be done not according to how you feel like and how comfortable you are and because you have little money. There have to be standards, and these standards are international standards that guide interventional studies or clinical trials. And I'll tell you, this came because of very, very terrible experiences that didn't happen just in our in just one African country, but they happened at a global level. And so the International Conference on Harmonization, ICH, it gives guidance on technical requirements for registration of pharmaceutical for human use. Drugs are going to be used for human use, bringing together drug regulatory authorities and pharmaceutical industry in Europe, Japan, and the United States in order to prevent duplication of trials and also to ensure that, they streamline the regulatory assessment of new drug applications. Of course, you will find a lot of drug, and you say, but for me, I'm in medical devices. And that's the thing. The drug in the pharma industry has really progressed for a long time because much of the first interventions that we're looking for were in curative. But now there has been a lot of technological advancement that has followed that we now look for technological treatments, diagnostics, and all these things. So ICH still is a guide for medical device trials. There was a lot of issues that led to and I could say disasters, that led to to the developing a standard ethical code that guide research. There was, for example, the syphilis trial, which was done in the US, and which exposed African men, black men, to untreated syphilis for over 40 years, under the disguise of them getting free health care. So they would come, but they are not treated, even if penicillin had been discovered and found to be effective towards treating of syphilis. So they kept coming, and they are not even knowing that they are being followed up. And many died of syphilis. And until when this leaked into the media, newspaper, the New York Times, and they talk about the victims which have suffered unknowingly. The same thing was the thalidomide trial. Thalidomide is a drug which was being given, and they were saying, okay, it's to help to sleep, it help you to sleep. But it was given to also pregnant women and lactating mothers. And for 12 years, babies were being born without limbs. And there were all these effects, but people were quiet. So out of this, it was agreed that no, there should be some guidance. Trials are good. They help to generate good solutions that can solve human problems, but we need to put some form of ethical standards and controls. So the Nuremberg Code came up, and those are very, very key areas. And that is the pipeline really which happened. In 1947, the Nuremberg Code came up, and you see this was after World War II. And these research ethic principles for human experiments, especially after the Nuremberg trials had happened, started off the ethical line. And over the years, we had many others, the World Medical Association Declaration of Helsinki, which we still submit to as we implement trials, and then the Committee of Safety of Medicine in the UK, and then the National Commission of Protection of Human Subjects of Biomedical and Behavioral Research, the Belmonte Report, very important for you to read them, those reports, and then the Good Clinical Practice, the US Food and Drug Authority, which generated, and we still follow this GCP. Of course, it was updated, and there was also Good Clinical Practices Europe, and now we said, okay, now they said, why have GCP of Europe and also have GCP of Good Clinical Practice of United States? So they said, let's harmonize, and that's the international harmonization of these guidelines of good clinical practices that had to come out. And I want to invite Susan. She's going to tell you a bit of the role of good clinical practice in medical device trials. And these ICH guidelines, they have both the quality guidelines, the safety guidelines, the efficacy guidelines, and multidisciplinary guidelines. And you'll be able to see that. So in this slide, good clinical practice, Susan is a clinical trial monitor, and she has been in this for quite a long time. I want her to briefly, just as I make this pause, for her to take you through the 13 areas that make up the ICH GCP, good clinical practices. And it's a cycle. I would need you to talk about it, and I think those who are online, you're able to see it. So they start with number 1, number 2, number 3, number 4, and they go round. And how does this Susan, come? How does these principles of GCP apply and in a medical device trial, and what do they help? I know you're always checking out that. You're always checking out that. So maybe you can take people through that and give some explanation.
Okay. So I'm going to take you through the principles of GCP and why they should be applied also in other trials. And why he said, briefly, he said that every trial we are doing, whether diagnostic, whether device, whether vaccine, we are going to do it in a human being. And the reason why these principles came up is to standardize, because at the end of the day, when these products are licensed, they're going to be put on market. If it was done in Uganda or Kenya, wherever it's done, or Asia, it should be done in a standard way, because it's going to be marketed at one time. So one of the things we look at the ethical principles, meaning these medical devices. Today we are concentrating on the medical devices. I'm not going to talk about drugs and what. But they should meet what we call the ethical principles. And in this, the purpose of the ethical principles is to avoid what happened in the past. So that as a researcher, your aim is to protect the human participants. And the next that we are going to look at is the trial risk versus the benefits. As a researcher who is looking at this device, you have to ensure that the benefits outweigh the risks. Like the safety trial, you see the risks were outweighing the benefits. And the next, you have to look at the safety, the rights, and the well-being of the trial participants as a researcher of this medical device. And this doesn't apply only to drugs, but also to devices, meaning something may go wrong. So you have to put this in place. Then the next, you should have information on this medical product prior for all those stages he has taken us through, meaning each stage you went through, there is that information you accumulated, and this information should be available to the researchers working on this device. Then secondly, we have to look at what we call good quality trials. Remember, we put several measures in place as researchers of these devices. You should have SOPs, you should train the staff. At the end of that day, when they come to audit you 10 years, 15 years from now, to see that you really passed the processes. Then we also talk about compliance with the study protocol, meaning as a researcher, you have to put several measures. So you'll put measures. Who are those people who will ensure that you comply with the protocol? Maybe the regulatory bodies, the research ethics committees, and other bodies. But internally also, you may have what we call an internal monitor and others to ensure you comply with the protocol that was approved by these bodies. Then also we talked about medical decisions, and these medical decisions should be done by a qualified physician. That's why we always remove what we call conflict of interest in research, meaning the PI is qualified technically, but you have to employ study doctors who have the experience, the education. So for us as monitors, when we come on your site, we'll look at the documents where is evidence that shows that your team, especially the physicians, are qualified. And why are we talking about physicians? These are people who will be able to ensure the safety of the participants who are using this device. Then we'll look also at the trial staff, meaning all your trial staff who are working on this device have to be qualified by education, experience, and this should be also documented, like we said. Then whoever is going to participate in your study of this medical device has to provide an informed consent voluntarily. And you have to ensure that we are trying this medical device. And this informed consent should be also documented. And it should also be translated in a language which the participant can understand, and it should be a layman's language. Then we also look at the clinical trial data. And like we said, it should be recorded and handled in a proper way. And before they do recording, that's why you see in studies, you have to ensure that team who are going to collect this data, that they are trained. They understand the data collection tools. And they are trained over time, and this is always documented. And you should always have what we call meetings to see how is the data being collected before it's entered in the database. And also what we call quality control checks. And you should always have people who can always, like statisticians, who can do what we call interim analysis to give you what is going on. So we'll talk about confidentiality. Remember what we are doing it in human beings. So we have to ensure confidentiality of the participants, but also the data we are collecting, whereby we don't use their names, we will use codes, but also to ensure when they are participating, there is confidentiality. No one should know that I'm participating, but you should assure me of that confidentiality. Then we have talked about good manufacturing practice. It's a requirement. If your product or device is going to go what we call post-market, it should have passed through what we call good manufacturing practice, and they should also be document. And lastly, there's what we call quality assurance. Remember, you are doing this device, but you are not the only manufacturer in the country. So if you do not participate through what we call quality assurance, and this is a responsibility of the sponsor, and he will take you through the various responsibilities of other people. So I'm done. Thank you.
Thank you so much. Let's clap for her. That was amazing. You know, she speaks with authority because that's what she does every day of her life. And thank you so much, Susan. So that tells you that clinical trials really require a lot of robust process. And we look at different things. Now, I want to you have a nice device, you have got your permissions from the Minister of Health or NDA. They have classified it already. They have classified it, whether it's class A or class B, C, D. Let's say it is class B and above, meaning it's invasive. It has significant risk to human being. Okay, so you have also made sure you have registered it to get an IP, or like a copyright, to avoid that when you begin exposing it out, that someone steals it. So the next stage is that you're going to now engage in the preparation of the clinical trial, meaning you have to know who can help you to design a proposal. You have to get a contract research organization, which can be a person, it can be a group, an organization that can take up the investigating of your product, because you can't do it yourself. That will be a conflict of interest and it will never be accepted. So you want now to move to that stage. And of course, why you want to move to that stage, you may still not be having the money. You need to mobilize the money for doing that clinical trial. So where do you start from? You have to come up with a trial synopsis, a trial summary. And this is a simple, you could call it a concept in the normal language, but we call it a trial synopsis. So it has a background and the rationale and the objectives of the trial and the methods in simplicity. And it can be 2 pages, 3, 4, but not so huge. And this is what you used to discuss with scientific experts, and you use it to identify partners to work with and sites that you will implement this trial in. And it can be used also for fundraising when you're looking for funds. So you use a bit of that synopsis. You don't first develop a whole proposal and then you look for funding for this. You first develop a synopsis and you use it to get partners to engage with different funders. After that, let's say you've known your sites, you've known whom to work with. So study protocol has to be developed. We call it a study protocol. I know people are used to proposal. Well, in trials, we call it study protocol. Now, you develop this after you have partners and the sponsor is identified. Sometimes when you get the money as an innovator, then that person who has given you money is the funder. But now you are becoming a sponsor. You're becoming a sponsor. You can either be a sponsor or you can also delegate the sponsor roles to another person or an entity. But you can be a sponsor. And then you subcontract a contract research organization or another person who will now implement or design this trial and run it. So they will be the principal investigator of that trial. You are the developer of the device. They are the principal investigators of the trial. And they independently implement this trial according to standard processes, as we will talk about them. So we have finished 2 stages. The next one is site identification. What do you think about when you're thinking about identifying a site where to implement your trial? You should deal with a site who are concerned about the question you want to answer. Are they interested in that kind of trials? Next, what is the disease prevalence in that area? Can you get the participants that you need to enroll in your trial? And clinical trial experience. Do they have the experience to implement such trials? That you can sit back and say, okay, I know I've engaged an experienced team. Then interest from the research community. You also need to know what the research community is talking about. They may be interested in having trials that include Africans, include a certain group. So, as a gap. So, you want to listen to what the research community is also saying. So, it guides you in identifying. And when you're selecting, it's important to do a site assessment. There's a due diligence questionnaire that has to be developed that helps you to assess. And so you do an assessment visit to assess whether this facility has what is required to implement a trial. So meaning, trials can't be just be implemented in a wardrobe somewhere. The site is very, very important. And certain things being in place is very important. So the due diligence questionnaire is very important. And recruitment capacity, you assess the recruitment capacity. Are they able to recruit? If it's going to be in a hospital, how often do such patients come? It guides you on your timeline and the budget. Site organization, how is it organized? Won't my trial, things get lost in another trial? So the organization is super important. Look at the regulatory requirements. If it's a country, you want to do it in Uganda, how is the regulatory requirements? Will it be the same if you're doing it in Cameroon? Will it be the same if you're doing it in South Africa? Sometimes you go to a country, you do a trial, and 2 years pass, you have no ethical approval. And even the funder wants their money back. So because you didn't do a proper site assessment, there should be an environment that supports that kind of trial at a national level. Feasibility aspects, check whether it's feasible. Can you get the right human resource in that setting? Do you have access to the technologies that you need? To the diagnostic? If you're dealing with patients, if they get injured in a trial, is there a facility where they can be taken to get the best care? So that takes us to trial implementation. Let's say all that is done. You have it sorted. Then, how are we implementing this trial? There must be a research team and partners. Clinical trials cannot be implemented in solitary. There must be the strength of implementing a clinical trial in the right way. It is in working with other partners and other people who are able to look in there. And we shall see that. Anybody involved in a trial should have the knowledge, training, and expertise required to cover his or their tasks. I think Susan mentioned it. Very important that whatever role you give a person, you have to be sure they are trained and it's on record and that they have the certification that shows that they qualify to do that particular task in a trial. Less of that, you have not followed good clinical practices. Then, two key actors are there when you think about a research team. The two actors are two. Sponsor and investigator. And we shall see the role of a sponsor and the role of an investigator. But you need to know who is going to be the sponsor. It's either you or you're going to delegate. And who is going to be the investigator, the one who is going to lead the research, the clinical trial. Those are the two you need to identify. And these delegate their tasks to everyone else, and we shall see whom do they delegate to. But these are the two. So, sponsor. What's the role of a sponsor? It can be an individual, a company, like Soil Research Foundation. It can be a university. It can be a sponsor. It can be a company, your company. Meaning, what do? They take responsibility for initiation, management, and or financing of a clinical trial. So their work is to make sure that this trial is initiated well, it's monitored well, it is managed well, and it is well financed. Whatever is required is provided. So meaning you may have a funder, but then you have a separate sponsor who is going to work out the trial itself. Now, their focus is on respecting of the protocol. A protocol which was developed is being respected during the trial implementation. So they will engage a clinical trial monitor, like Susan, to come on their behalf to check how the trial is being implemented and give a report. They will check on the respect of ethical principles. Is it being implemented according to ethical principles? What is the safety of participants? Are they safe, or we are getting many problems? Quality of the data, data integrity. They will look into that quality of the data, because research is about data. If the data is fake, then the results are what? Are fake. They are not good. They are not good at all. And meaning people will make wrong decisions. They will pass wrong guidelines. And the human problem will continue. So they will look at adequate analysis of the data. They want to be sure that the data of this trial has been analyzed well, and that it is well disseminated, that before it is disseminated outside, it is the right data that has been analyzed properly. They can delegate their tasks to, and they can also delegate to a contract research organization, which can be a person. That person can be called a trial manager, or an organization like this one. It can be academic, it can be a company, it can be an organization, but it's contracted by a sponsor to perform one or more of the sponsor's trial-related duties and functions. The next important person is the investigators. Principal investigator, who is going to lead this trial? A person responsible to conduct the trial at the trial site is the leader of the team. Now, they have to make sure that, again, all the team members, the trial team, don't deviate from the protocol that has been approved by the ethics committee and by the sponsor. If we agreed on a protocol, that's what should be implemented. They also delegate responsibility. They can delegate to sub-investigators, and they can fill what we call a delegation log. You delegate, but you document. Whom have you delegated? I've delegated to finance, to be the finance person on this trial. I've delegated to logistics. I've delegated to a social worker. I've delegated a doctor. I've delegated. So everyone else, they get their roles from a sponsor or from the investigator. The whole trial team, even a driver, they get their roles from these two. And at times we can have what they call a coordinating investigator. In case you have a multi-center trial and you need a small core team which can coordinate all those centers. So that person can be called a coordinating investigator, and they supporting the investigators at different sites. The other trial personnel that become part of the trial team, like a trial manager, the site coordinators of the trial, data managers, statistician, data entry clerks, pharmacist if it's a drug, biologist, nurses, counselors, biomedical engineer, has to be part of. If it's a medical device, you have to have a biomedical engineer too on the team. And data monitors, IT, and drivers, and you can mention even cleaners. All personnel should have knowledge, training, and expertise to perform their duties, and that's a key point. And training on good clinical practice is a must. They must have gone through that training, and it's a free training. You can get it online. Good clinical practice training, and no one works in a trial without that certification, and they must follow the study procedures. We set up trial committees. when we are starting a trial. So it's not about investigator having a protocol and a sponsor. No, there must be other observers who check the quality and the way how this trial is implemented. The trial committees could include coordinating committee, which is organized by the sponsor to coordinate the multi-center trial. If the trial is in many centers, the sponsor can say, I think I need a small committee with me, which coordinates all these sites. Then at time we can have what we call a data safety monitoring committee. A data safety monitoring committee, this is a must. It must be on every trial. At time we call it independent data monitoring committee. So it is normally established not by the investigator, but by the sponsor, to assess at intervals the progress of the trial. And they will look mainly on the safety data to ensure that participants' safety is assessed. They will even do what Susan mentioned. They will ask the statistician in the trial to do an interim analysis to try to give results by arm to try to see if there are more adverse events on the intervention arm compared to the standard of care. And if a limit was set in the protocol of when to stop the trial, sometimes we set that limit and say if we get 10% of this group get adverse events of this grade, we terminate the trial. Sometimes we can terminate the trial or we can terminate an arm. Just one arm if there are multiple arms. Then they also look at the critical efficacy endpoint to see if it's working. There are trials which never ended, like the penicillin trial didn't end. One of the trials, it was a polio. The polio trial never ended because suddenly when they began giving a vaccine of polio, they found that the group which was receiving the vaccine, they were protected, while others were not. So they said, now why finish this trial? Let everyone receive. So they did mass vaccination because everyone was suffering with polio in the US. Even the president of the United States then had got polio. So it was urgent. So this data safety monitoring board, it does that role, and it has external members only. It has external members, and it follows a predetermined charter. They develop a charter that guides the way they run and how many meetings to have. They meet. When they meet, they have an open session where the principal investigator also attends. And then there's a closed session where the principal investigator is not allowed to attend, but the biostatistician attends. That's where the biostatistician shares that data by arm on safety and efficacy, even as the trial is going on. The principal investigator must not know those results. So the statistician does not share with the principal investigator. The principal investigator remains blind to the results where the trial is still running until the end. So, and they access data by arm. The other committees that are set up and which support the investigator is the Trial Scientific Committee, which we also call the Scientific Advisory Board. Now, this has to be set up by people who have experience. It's both external and internal members. Even the PI can be, principal investigator can be part of this. But they get people who are good in that discipline, who have experience in clinical trials, who can contribute or guide in a scientific discussion of how to interpret the results of the study, how to implement it, how to decide when things have gone wrong. They are the people the PI, the principal investigator, relies on to give them advice, to give him or her advice on how the trial can be implemented. And they never access results by arm. For them, they are there to support the principal investigator, and the principal investigator has a role in setting up this team, unlike the sponsor who sets up a DSMB. Other committees are community advisory board. Every trial must have a community advisory board. These are members picked selectively from the community that help to guide on first the planning of that trial, its implementation, and also at the end of the trial, when you have finished, how these results can be communicated to the public, and also how they can be translated. So they can participate in all those areas. Some trials can have a clinical advisory committee, meaning there is a lot of clinical problems. So you may need a clinical committee that can look into the adverse events that are being reported, so they can help the clinician and the doctor to make some decisions of how to manage the patient in a trial. Sometimes we have an event validation committee, meaning some events have happened, but we are not sure. We need a team. There is no clear algorithm to guide decision making on whether this person has a condition or they don't. So sometimes you want to put up a team which can validate the events you are reporting, and they will check them and say, you said it's related to the drug. No, I think it's not. So they're able to guide you and in validating events. Terms, if you are storing samples, there will be a biobank committee. This, of course, deals with stored samples, and samples at times are kept for a very long time. Now, when you think about trial implementation, teamwork, it's about teamwork. It's about teamwork. You work with different people. Just to start with, just to run through, always, number one thing, if you're going to implement, get approvals. Get approvals, ethical approvals, National Drug Authority, and the ethical entities which are in your country where you're going to do it. Think about administration and finances. This signing contracts with the service providers, the PI institution, the contract research organization. There will be contracts, staff recruitment, very important. Trial budgets have to be made, and the grant management. There should be a system to manage funds that are for trials. And logistics, there should be an IT, there should be a transport system. You have to think about the supply. If you are importing things from other countries or you're doing local purchase, how are you going to organize the supply? How are you going to check quality of the products you're going to use? And pharmacy, do you need a pharmacy? Think about the procurement process of the drugs, the labeling. Everything that belongs to trial has to be labeled for clinical trial use. For clinical trial use, so they don't get mixed up with other things you're keeping in the store. Blinding, think about blinding. Covering things so the patient can imagine it's the same. Okay? And stock management, very important. Susan has mentioned the clinical monitors. When they come, the clinical trial monitors, they are looking in the pharmacy. They will count drug by drug. They will check how much has been dispensed. They will check the stock management system, and even destruction plan, how to destroy the investigational products after the trials. Some things are for testing, and after the balance is to be destroyed. Even the destruction plan has to be there, and we must know where they will be destroyed. We must have a certificate of destruction proving that they have been destroyed. And so all those are important things. Data management, think about data. Data is key. And where per used to questionnaires, here we talk about case report forms, CRF, case report forms. So a clinical trial can't have one document called questionnaire. No, it's not questionnaire. It's case report form. And there are more than one. They can be 10, 20, 30 per person. Per person, depending on what you're monitoring. And there should be also a CRF, or case report form completion guideline. How should people feel that? Those forms, there should be a guideline developed. There should be a data management plan developed before. And statistics, there should be a statistical analysis plan. Now, this analysis plan at times is developed after the trial has been approved. And once it's developed, it has to be discussed by the trial scientific committee for agreement, and the sponsor to see if that is the right way this result should be analyzed. Because poor analysis, wrong reports. Yeah. And the randomization list, you have to think about it. Normally, the statistician develops the randomization list, because poor randomization affects their analysis. So they develop it. And when they randomize, they can give you maybe slips in opaque envelopes so that you can, once a patient comes, the nurse just opens and then knows which arm this person is without influencing their choice. And look at training. No, quality. Quality, she had mentioned also standard operating procedures. Everything in a trial, every procedure must have a standard operating procedure. There's nothing obvious. Even if it's taking a temperature with a thermometer, we have to see an SOP. And that SOP has to be well written, reviewed by the right people, and validated and verified. Then quality management guideline has to be there to guide the clinical monitoring, which has to be done during the process. But also, if you have to do some bit of quality controls, calibration of the device, how is the calibration going to be done, how often, and all that is very important. Do we have the right person to do the calibration every day, troubleshooting, and all that? So we have to think about that as we're planning. Training, everyone should have good clinical practice. They should be trained in all the standard operating procedures related to the tasks they're going to do. And there should be a training plan. And all trainings must be recorded and documented. Must be documented. Safety. We have to have a safety manual and a pharmacovigilance plan. How do we record adverse events? How do we report them? And how do we manage them? So all that has to happen. And documentation and documentation and documentation. That is what makes clinical trials. Everything must be documented, whatever it is. So we have to have a trial master file. A trial master file has all the essential documents. And we shall see some of them. But that trial master file is always with a sponsor. And we also have site investigators file, which have to be on every site where the trial is being implemented. They have to be that site investigation file. So the documents in both are similar. But it's just that, wherever the trial is, there must be documents. So that when the regulatory bodies come to audit you, they find them there. Now, when you're doing trial preparation, just to run quickly, I've mentioned the summary of them. Please seek approval from all ethics committees. If you have chosen a country we're going to implement the trial, if it's here in Uganda, understand the country's ethics committees. At times they are institutional ones and then the national platforms. So like in Uganda, we have the research ethics committees at institutions, and we have also the Uganda National Council of Science and Technology. And also, you have to follow the guidelines that they give. And at times they need a protocol, the investigators, CVs, and informed consent at a minimum. But you have to follow the national guidelines. Study insurance. Patients have to have an insurance. You have to buy for them an insurance. Even the investigators have to be protected. So there should be an insurance to protect the investigators. So that's what makes it costly, because there is all that to do. And the National Drug Authority or any regulator of medical devices, you have to get to know what they need. And you have to notify them, you have to submit to them. You have to engage them early. Actually, the National Drug Authority, for us in our country, you engage them before you start the protocol. Because they are the ones to classify for you your device. And then you engage them during the submission of the protocol at some point, and even after, to give you permission to go to market or to register. So the trial preparation and authorization and contracts, they can be at the site level, or at times they are corporate agreements. You have worked in a consortium. People need to agree, and they write those agreements. They have to be on file, because all that is very important. Confidentiality agreements. Everyone who participates in a trial must sign a confidentiality agreement. And budget, please, a trial budget has to be detailed to cover a lot of lines. that are very important. So salaries, costs of tests, maintenance, drugs, equipment, consumable, facility costs, all that. Insurance, you don't forget all that, including archiving. And management of safety event. If a patient gets a safety event, they will need to be taken care of. So you have to have an envelope to manage their health care while they are still in a trial, because we can't be sure that what they are suffering is not from your device or it's not from the drug you're giving them. So that's very important. Materials, think about the study products that are needed, their availability, the laboratory tests. Are you engaging different laboratories to manage the lab tests, just in case one doesn't have the service or their machine breaks? Which backup do you have? Transport, think about shipment of the materials you need, if they're international or versus local purchases. And think about security of the trial site. Storage, documents, how are they being stored? The samples, how are they being stored? Internet access, because you want to communicate. And now we go to the investigational product or drugs. When we talk about investigational product and drug, the sponsor has a role. They have to make sure that they are getting this investigational product from a company which is approved. It should be a quality approved manufacturing source. So if you, and that's why before we go into trials, we have to be sure that the device you're dealing with is of good quality. You followed all the guidelines that are required, the different ISO guidance, and you are following the quality management systems, and you're making sure that, you know, the regulators have also looked into your device, and they have given you also their guidelines, and you've given them attention, and you have documented the whole process. So that is very important at a sponsor level. They have to make sure that the certificate of analysis is there, the procurement documents are there. Is it a donation or it's procured? How? Labeling, things are labeled for trial, use only, and transport is in good conditions. That things are not messed up during transportation to the site. Site level, we do stock management. Where the trial is done, we do stock management. We make sure things are stored well. The drugs or devices are accounted for, and that the procedure of handling them is very clear, that they are not mishandled. And data and randomization and blinding, also it's a role of a sponsor. They have to make sure the data management plan is in place. All these documents are in place, the case report form, the database, the randomization list, the blinding procedures are clear, and at the site level, we check on the materials that can allow safe data collection, and the randomization is being done well, and there is good storage of data. There should be servers, there should be backup of data. We must not lose data at a site. And communication access in case of central randomization. Sometimes randomization is not done at the point where the patient has come to the nurse. Sometimes it's done, you make a phone call to a person, and then they randomize and they tell you which arm. So now imagine network is off. So we have to make sure that the connectivity is very good, that it doesn't slow central randomization. And we look at the data clerks. We have to make sure they are well trained and they can enter data properly. Quality management, again, the sponsor makes sure that the SOPs are all detailed and they are approved. The sponsor has to sign on every SOP. And that the clinical monitoring plan is in place. They have planned to send a monitor. They have planned to do some monitoring of the trial. And that the quality assurance is also in place. The site also ensures there's monitoring and site-specific procedures that help everyone to do their work well and to document things properly. And safety is a key thing as we implement trials, and the sponsor makes sure that there's a study insurance that covers participants, and that there's a system to handle serious adverse events or some suspected unexpected serious adverse reactions. Sometimes the sponsor sets up a pharmacovigilance unit dedicated to the trial. And that unit receives all these reports of safety that are coming from different sites. And they look through them, analyze them, and they guide the team on the site on how to manage them. And times the sponsor will set up a safety manual for notification of serious adverse events, and also SUSA and pregnancies. So pregnant people, once someone gets pregnant and they're taking a drug, or they have they're under an invasive thing that people that scientifically we think it may affect their pregnancy or the baby, that person has to be followed up even beyond delivery for the whole period of pregnancy and after delivery to confirm and assess the status of the baby. So the Data Safety Monitoring Board is a very key board that the sponsor sets up for safety monitoring. And at site level, the clinical investigators are trained to ensure that they recognize adverse events, document them well, report them well, and manage them well. Documentation is what I talked about. They are essential documents, but trials are about documenting. Every version of protocol that you're using, it has to be signed. It has to be the approved one by ethical committees. And all versions, if there's an amendment in the protocol, all that has to be kept. Every change in any document has to be tracked. Versions have to be tracked. We must know what the current version is. If a document receives a new version, all the old ones are removed from the circulation to avoid errors. And administrative and regulatory documents, approvals from ethical committees, everything has to be put to file. Study participants, their case report forms, their consent forms have to be stored. Remember, consent forms have to be two. One that the investigator or the nurse remains with, which is kept in the trial, and one other signed one has to be with the participant. So very important ethical practice. Products, they should have a certificate of analysis and importation authorization, and all that has to be on file. And there are a number of documentations, as I said. Some are essential documents, others are the trial master file and the site investigator file, and those are very key things. Investigators have a task of ensuring that there is respect to the protocol, that as their team is implementing the trial, they are following the protocol, they are following the standard operating procedures, they are respecting the good clinical guidelines, and the informed consent are well signed, and there is recording and notification of adverse events to the sponsor. The sponsor has to be reported to all the adverse events, and the data collection is moving on well in respect to the good clinical practice. Trial documentation is being done, and that the tasks have been well delegated, and the delegation is well documented. And that's very important to have. And quality monitoring, we normally have to have a site initiation visit. Before a trial is implemented, the trial monitor will come and assess the readiness of the trial site to ensure that it's ready to implement that trial. So that site initiation visit is done, and the report goes to the sponsor. So the sponsor gives the go-ahead that now let the trial start, basing on the report from the clinical trial monitor. Then there will be regulatory monitoring visits in the interim as the trial is running, and the clinical trial monitor is monitoring certain things, as we said, that the sponsor is interested in to ensure the trial is moving well. After the trial is done, there will be a site closure visit. And again, the monitor is checking how the trial has been done, whether proper documentation has been made, whether there's still something more to be done, like maybe the investigation products are not yet destroyed, they need to be destroyed. How do we close out, how do we store and archive the source documents, the case report forms? How do we, so they acquaint themselves in that, and they give a report to the sponsor, and the sponsor again has to say, okay, now the site is what? closed. The trial has been closed. So the delegation is done by the sponsor to the trial manager at times, or the monitor, who will respect the good clinical principles. They will check on the respect of good clinical principles and respect of the study protocol. The Data Safety Monitoring Board, we had talked about it, and they normally have their first meeting before the trial is done, starts, and they will review the protocol, and they will also review their charter, the Data Safety Monitoring Board charter, which is going to guide their meetings. They normally would meet 3 to 6 months interval. So regularly, 3 to 6 months interval, and they will have an open meeting, as we said, and they have to listen to the progress report of the trial and the safety review. They want to listen to every incident of safety, one by one. And they will also analyze it by arm. And they will also have a closed meeting to look at by arm. And they will also make sure they report to the sponsor and the investigators. Once the Data Safety Monitoring Board has met, the meetings and the decisions are also submitted to the Research Ethics Committee to say, hey, look, they sat and they said the trial should continue. Okay, so that's important. Then the Scientific Committee, as I told you, they support the investigator. They approve the study protocol. They look through and approve it before it's submitted. And even if there's an amendment, they will assess the amendments you want to make, and they will tell you, okay, but change this, but not this, right? So they guide the investigator. And they can also propose and review sub-studies. If you think you're going to do some sub-studies in a clinical trial, they want to listen, and they can also propose. And they also review the study progress. If there's any major deviations, they want to see that. And if there's any safety, they want to review that. So with that, as we come to the end, the communication and dissemination of results from a trial, it's another very important part of the clinical trial. The communication plan has to be agreed on by all the participants in the consortium. And if there are different centers, the principal investigators have to agree how are they going to publish the first paper. It is on the primary outcome. Okay, before that one goes out, there's no other paper of a sub-analysis which can go out. They will agree who will be the first author, who will be the authors, and who will be acknowledged. The tools to use. Remember, when we're disseminating, we think about different audiences, scientific audiences. Are we going to have international conferences? Are we going to do stakeholder engagements? Are we going to disseminate to patients and community? We want to disseminate all of them. And the methods can vary. And please, it is now part of practice and standard that there should be dissemination of clinical trial results to all these groups, scientific audiences, stakeholders, and patients, and community. So there should be a very well-designed community engagement plan that will cover all these elements. And the tools to use, the tools to use, it may be through reports, you may do meetings, publications, media campaign, websites, social media, leaflets, newspapers. You can use as many, depending on what you want to do. But everything that is related to dissemination must be first approved by the sponsor. You don't just make a presentation and go to a conference nearby to present a trial results without approval of the sponsor. So this is very important. And there should be a budget. In the budget, there must be a line for community engagement for all these things. And so the final trial report has to be given to the sponsor and to the funder. And there can be also scientific disseminations. Good to plan that. And dissemination to stakeholders, as we said, and communities. As I end, I always acknowledge the people who I recognize who have mentored me in clinical trials for over so many decades. I'm making almost 2 decades in clinical trials. And these have been the people who have trained me. And why I bring this slide is because mentorship is very key if you're going to perfect in your conduct of clinical trials. You have to learn. There's a lot of, practices we learn from different people and from different settings. So I've been very privileged to work with really high-profile researchers in the world who have taught me a lot, and which I'm very happy. And that's what inspires me to give such a lecture, to share the skills that I've learned in many places. So thank you for joining us. And that email there, you can always send an email there. And in case you want to ask about future research trainings and also other trainings which we offer. And please make Soul Research Foundation your number one choice as a site for your clinical trial, designing and implementation. We are also happy to serve you if you are interested. We'll be happy to work with you and to support you on all the process and to navigate the waters of regulation which many struggle with. And I just wanted to mention this, that our next one will be, our next lecture, we will tell you when it will be on the step-by-step process and requirements for drug medical device trials regulation approvals in Uganda, but it can be in any other place. And we'll be helping someone to know what is needed and walking through the process and having very amazing discussion. I hope I'll be able to have some people from the regulatory who will be able to also speak and tell you from word of mouth what is going on and how things should be done. So with that, I think I want to appreciate you for waiting. We delayed a bit and we went a bit over, but I hope it has been helpful. I would request now to go to questions and answers, and I will welcome Susan here. At least let's have 5 minutes or 10. Are there some questions, Caroline? All right, there are some questions. So we will sit and be able to receive these questions and be able to answer the people. And if you have other questions, if time catches us, please just go ahead and send an email, and we will try to create another broadcast, perhaps to answer them one by one in case there are many. And besides that, I think we had wanted people to open their videos, especially those who are online, so that we can have a snapshot and know who has been there, and want to appreciate you so much for this patience. And our hope is that this has been helpful to enlighten you of the process. We will be having a clinical trial series and module and program coming in the future. We'll be informing you, and you will have an opportunity to participate in all training from beginning to the end, so that it can help you to improve your expertise and skill in implementing trials. All right, so are we able to see the people on the screen? Okay, you tell me when you're done. Yeah, so, all right. Okay, so I stop sharing. I stop sharing, yeah. Okay, so I think we have questions which were sent before. Where are the questions? Are you going to read for us the questions? It will help us to... Oh, it's okay. So let's first open our videos and have a photo. Have a photo. This is a good way of having photo, right? I also put mine or something. Eh? It's okay. All right, good to see you. Yeah, give us those smiles, please. Yeah, good to see you. Good to see you. We are a big family, and we are all working to cause that change. And I'm glad that you have rejoined us today. Yes. All right, all right. The pictures are coming. Thank you, thank you, thank you, thank you, thank you. Just give us the pictures, and yeah, and then we shall have the questions. Yeah. Okay. Some more people giving us some faces. Okay. Well, you can take. Others might be in a very difficult place. Yeah. So really thank you. And now we are going to receive some feedback from you. There is a hand there. I hope we can increase the volume in the system and we hear. And then we can have others. So perhaps we can remove this one. Yeah. 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 So. All right. So maybe you will push the camera down. Thank you. Huh? All right. So, are we able to hear online? There is Mr. Ezekiel. Yeah. Ezekiel. So, are we able to get the sound through? All right, there's a heavy storm, or I hope it doesn't go through the microphone to disturb your hearing. but let's hear from the question. Are we able to hear, please? Sound. All right, they are trying to sort out the sound. Do we have some other questions which you can be giving us? Yeah. You may need to come closer.
Thank you very much. So the first questions are coming from Quizera. Question 1, she says, she's asking, most of the medical devices being developed in Africa are only improvement of the devices developed elsewhere who have undergone clinical trial before being allowed to be released to the market. My question is, do we need clinical trials for new devices which are an improvement of the existing one? If so, do we need to do all those phases of clinical trials?
All right, wonderful. And that's a very good question. We shall answer one by one for purpose of capturing answers properly. So, if it has been an improved, I'll give you an example, even in the drug or in the pharma trials. You can repurpose a drug. Just repurposing it to use it for something else. It's the same drug, it's approved, it's on market, it's licensed, but you are using it for a different purpose that is outside its licensing. Outside its licensing. So if that happens, that drug has to go through clinical trial for that new purpose. And it may not go through phase 1, but it will start from phase 2. It can't go to phase 3 straight. It has to start from phase 2, where you target that particular patient population which you want to address. So you don't go through all the phases, but you still go through clinical trial, but from phase 2. I hope that is clear. So it's the same with medical devices. You might use a device and then you have changed something. Or you have changed its design. You have you've developed a local prototype, but we still need to assess to see if it can produce the same outcomes like the original device. All right?
And also, in addition, you need to seek approval from the initial owner of that device before you redesign it or you revise it. You need to seek that approval.
Good. Next question. You may have to come closer if it doesn't kill.
How can we tell the number of participants required for clinical trials to be approved, and what does it depend on, specifically for medical devices?
So I think I gave a breakdown of the number of participants, the range, for each trial phase. So that guides you, number one. Number two, you will still calculate a sample size based on the outcome you are looking for in your specific trial. But the idea is that whatever the sample size you're calculating, it has to follow within those boundaries. So you can't say, for me, I calculated phase one, I'm going for 100. I'm going for 300. No. Huh? No. So that guidance gives you a limit to fall in, but then you still have to follow scientific methods to to power your study to be able to detect a certain difference, a clinical difference between the group which is receiving the intervention and the group which is receiving the standard of care. Next question.
Maybe just to add on also, like we said in research, we don't work in isolation. So sometimes we may engage a statistician to help you to know the power and so forth. So thank you.
There must be a trial statistician according to good clinical practice. Me, I'm a biostatistician. When I become a clinical trial principal investigator, I must have a trial statistician. The point is very simple. You've seen the data the independent data monitoring board. The PI, the principal investigator, doesn't attend the closed session. It's the statistician who analyzes, does interim analysis, and attends and shares the results. And the principal investigator is not even supposed to know those results. So now imagine you are the principal investigator and at the same time you are the statistician. You'll be stuck.
Question 3. When is the single-arm clinical trial required? Does it also have those phases, specifically medical devices?
Now, times we use single-arm trials, and normally when we are validating simple devices, which might be class A, the non-significant risk devices. So you are just assessing its impact in a pragmatic setting or in a community. So meaning you may do a before and after assessment. You assess before, then you do the intervention, and then you assess after. So even in that case, you are comparing the effect of that device in the same participant. We can also control that and do a controlled before after. Controlled before after intervention study, where you get one group where you give the intervention, assess before and after, but also get another one which is receiving standard of care. But in such arrangements, you are not dealing with just individuals, you are dealing with groups of people as an experimental unit. As an experimental unit. So in such, we can have a single intervention trial and single arm trial. But of course, it will be, if it's not controlled, there will always be questions of how sure are we that all the effect we see is a product of just the intervention and not something which would have happened even without the interventions. So that's why we need a control many times. Yeah.
As per, this is AIGA, as per the contest of types of clinical trials.
Yeah, so I don't know whether you are able to hear. Let's hear from in the chat if you are still able to hear, because we have a heavy rain outside. I don't know how it might be coming through. So you'll forgive us. Yes?
Okay, so the other question says, as per the contest of types of clinical trials, assuming I have an AI model to improve patient diagnosis, what phases of clinical trials can it undergo before you roll it out to the market?
So it is an AI-based algorithm
Yes.
for diagnosis, meaning you want to do a diagnostic kind of trial. You're going to compare. AI is a means. So you're using the AI algorithm to see if it can help you to identify people who have a disease. Again, you will have to compare it with an existing standard of care to be sure that it can detect as good as the existing standard. And maybe you will now gain some benefit on the efficiency, that maybe it is faster, it doesn't need a lot of human engagement, and maybe there are less chances of subjectivity and bias in diagnosis. So such a study, again, you have to submit to the National Drug Authority to they have to be notified, because they may have the mandate over every clinical trial. So even if they may not supervise your trial, they want to be notified that you are going to do a clinical trial. And that, in my view, it will not be a significant risk. It will not be a significant risk trial. It is a non-significant, a low risk trial. So you'll be able to do perhaps you may start from phase 2 and start, as long as there have been studies that show that this AI has been validated in human beings. So the first thing is validate it in human population. If they have been validated, you have checked that it's sensitive, it's good. Now you are moving to a point of comparing it to the existing. Then you may use that scientific rationale to now move to phase 2, to start on the second phase and subject it to a bigger population of participants and compare it head on with the standard of care. Yeah, I hope I've answered.
The next question is, what is the estimated smallest budget for a clinical trial?
What is the smallest estimated budget for a clinical trial? Someone sent me a WhatsApp yesterday and was asking me just that question. And look, what we said, clinical trials require money. And we can't just get an amount out of our heads. But what I can say is that the cost of a clinical trial is driven by the regulatory requirements? Does it need a full supervision of the National Drug Authority? It is driven by the materials you need, the kind of human resource, the follow-up, how long is the follow-up, the number of patients, participants you're going to recruit. It's going to require, do you need laboratory investigations? Laboratory can take more. Which more investigations do you need? All that can push up the budget of clinical trials. And licenses. Licenses. The insurance, the patient insurance can take around 5,000 USD and more. 5,000, 6,000. Just patient insurance, depending on the number of participants, if you have like 30. So the cost is high. And ethical approvals, like in our country, if you're going to go to the National Drug Authority, you need 4,500 for a trial, US dollars. And you need 300 for Uganda National Council of Science and Technology, US dollars. Research of this committee, you need 300 US dollars. If there's an amendment, you're going to need some more money right there, just in regulatory. There are meetings you're going to have, meetings, pre-meetings, to get feedback from different entities about your device. You need to budget for that too. And so there are some costs which are in just regulatory. Before even we talk about the setup, the site setup. And that's why I told you that centers like Soil Research Foundation, when we have set up a setting, it allows an investigator to come and implement without reinventing the wheel. So some costs go out. But if you are to set up everything, the costs can go high. So it is on trial basis. I think we have to look into the trial to be able to assess. But I'll tell you, phase 1, it is good. This is a off-head cost I gave someone. Think about 50,000 to 100 US dollars. 50 to 90,000. And then phase 2, more numbers, more money. You may need to think about more than that. 100, 150. Phase 3, you are in 150, 300,000 US dollars. So, in my experience, trials go around that. Yeah. I really don't know whether you're still hearing because we have a heavy storm. They are hearing. Okay. Do you have another question? So, those were the questions. But Ezekiel had a question. I don't know if you can type it. We can answer it. We can hear him? Okay. All right. Ezekiel, you can go ahead. I'm trying to hear. I'm not hearing properly. Are you hearing?
Hello?
We are not. Yes, we are trying to hear you because we are competing with the storm outside. Sorry about that.
I will try to reach my voice.
Yes, you can.
Yes, so I'm actually asking, why should we separate the number 3 from number 9 in the 13 principles of clinical trials, where we have the participants and then the informed consent? Why can't it be one? Or did you do the arrangement in order of relevance? That is why you have to separate the participant number 3 and then consent number 9.
So you are talking about the GCP principles. Yes, are you able to answer?
So how I can respond to you that all the GCP principles have an equal weight. Meaning if you remove one principle, then it affects the other principles. It doesn't matter in the number of numbering, but but they all carry an equal weight. Meaning, if you are to do a trial, all of them should be in place. Over.
That's exactly that. We didn't organize them. They are all required, the 13. And if you are doing a trial, it has to be assessed on all the 13 principles of good clinical practice. Thank you.
Okay, the second question before I leave you is if the device is clinically tried and then accepted and it's on the market, how many years or how long does it take for recalibration of this device? Are you going to go back to send it, subject it to another clinical trial, or have a manufacturer that will require recalibration?
So, if you are recalibrating after post-marketing, you may not have to go back to a full-fledged clinical trial, but you may still do some validation studies to assess, to see if there is benefit, there has been an additional benefit based on calibration, or if there are some new safety issues related to that calibration. And of course, you need to update that information. I think also in your licensing, just to make sure that you give some updated information about your device and a new version. Yeah. But those will be validation studies that you can do post-marketing.
Thank you very much.
You're welcome. I think we are going to stop here, and thank you so much for having joined us. And may God bless you and continue to do the good work you're doing as innovators and as researchers. Thank you. Bye-bye.